Alright, so for the last of our macromolecule groups, we have nucleic acids. Nucleic acids are the things we typically think of as DNA and RNA, and so those are the two major categories. We find nucleic acids in a variety of places. We definitely find it in the nucleus when we're talking about a eukaryotic cell. We find it out in that nucleoid region in a prokaryotic cell. We can also, though, find nucleic acids within mitochondria and within chloroplasts. So these two organelles, the mitochondria and the chloroplasts, that are really associated with energy production in cells, have their own type of DNA, which makes it us suspect that these were actually independent organisms at one point and then became incorporated into eukaryotic cells. So let's take a look, though, at sort of this central dogma that we've talked about before. The fact that DNA, we know, is the code, and it's the code for all of the genes, which all of those genes are coding for proteins. So we have, within all of our DNA, we have a code on here in this double helix. In order for some of that code to get translated into a protein, there needs to be a copy. And that right here, we see that the DNA is going to get transcribed, in other words, sort of written, into a single-stranded form called RNA. Messenger RNA is really what this is, and it's going to move out of the nucleus and go to ribosomes and start to produce this primary structure of proteins. And then ultimately we end up with those proteins going through their normal type of folding, primary, secondary, tertiary, and quaternary in some cases. So this is kind of the general idea. RNA in particular, if we were going to break this down a little bit, we know that there's different types of RNA. And we have the messenger RNA, which we just mentioned, which is the one that actually transcribes a part of the DNA molecule so that it has that code on it. We have something called transfer RNA, which we'll look at pretty soon in coming chapters, which is something that's going to help to translate that code. And then we have ribosomal RNA, and this is the type of RNA that is going to be involved with actually putting those amino acids together. So that's ribosomal. So these are the three main types of RNA that we're concerned with. When we look at an individual unit, though, because we're talking about the macromolecules and we're talking about things that in most cases are built from monomers or individual units, the individual units for DNA and RNA are called nucleotides. And remember that a nucleotide has these three parts. It has a nitrogen base, or nitrogenous base, as sometimes it's said. It just means that within the structure is you'll find nitrogen. It'll have some type of a pentose sugar, so that would be either deoxyribose or ribose, as you can see below. And then it's going to have one or more phosphate groups. So the nitrogen bases are either going to be one of these, cytosine, thymine, in the case of DNA, adenine, and guanine. So these four are the ones that we see in DNA. But RNA replaces thymine with uracil. And these are called pyrimidines, and these are called purines. So that's the general structures that build these nucleotides, and then the nucleotides build what we call the monomer of nucleic acids. So let's start, we'll look at DNA. We know it's a double helix, and what we find is that sugar and phosphate part of that monomer form this backbone, this part that's like this part that's that the actual nitrogen bases are on, and then the nitrogen bases are in the interior. So this is a helix because it has this kind of a shape, and that the strands run in opposite directions. So on one side is anti-parallel to the other. We know that the bases or the nucleotides themselves join and form that backbone through something called phosphodiester bonds, which means that each one of these phosphates with the sugar on it are going to join in a particular way, namely this phosphodiester bond. We've looked at esters already, ester bonds in lipids. We also know that on the rungs of the ladder are formed by the base pairs. So we have thymine and adenine, which form they have hydrogen bonds, 2 hydrogen bonds between them, and then we have guanine and cytosine, which also forms hydrogen bonds between them, but in this case there's 3. So we see how that works. One strand is going to run what we call from a 5' to a 3' direction, and the other from a 3 to a 5. So this won't be, I won't put this on the exam this time. We're going to look at this again later on, but just understanding that sort of basic idea. So again, the code is in the DNA. It's going to get transcribed into a single strand of mRNA. It's going to then go to a ribosome where it's going to form this primary structure through linking amino acids by peptide bonds, and then it continues to fold until it becomes a functional protein. So lots of ways to look at that. We could look at a sequence of DNA, so as you see below here, AATT, GC, GC, and we could then know what the mRNA version of that would be with, remember that we normally when we'd see A on one side, we would see T on the other, but remember thymine is replaced in RNA by uracil. So here we have U, U, A, C, G, C, G, which is the complement to what you just saw up here, this half of this DNA molecule that is allowed itself to be copied. This would be that's copied. We spent a little bit of time already talking about mRNA sort of and rRNA and tRNA, so I think I'm going to leave this slide to go on to this one just to show you that this is sort of the assembly line that happens on the ribosome. So it's the ribosome that is this little structure. It's actually 2 little structures here. They're called subunits, and here is where the mRNA would kind of run through this structure, and as it was going through, what would happen is the tRNA, this transfer RNA, would actually bring an amino acid to the place where the next one should be, and that would form peptide bond, and then this thing keeps moving, and then we just assemble this primary structure. So again, we're going to see this again in more detail later. This table I pointed out to you before is just like comparing the difference between DNA and RNA. And then finally, the thing that I've talked about a few times, and this is just a nice little view of it again, is this idea that this is the set called the central dogma of life. And that is the idea that DNA has the copy to all of the stuff that makes us. But it gets converted into RNA, and that RNA gets converted into proteins. So we look at this idea of DNA forming RNA, but RNA then creating these proteins that build so many things in our bodies. So that's it on the nucleic acids. I think we kind of covered quite a bit there, and I will leave it at that.